But where I am aiming, that cortex and paracortex and whatever, it's a different kind of lymphadenopathy, and that's in case of lymphomas. Okay, and I want to tell you the difference. So, in case of, well, of course, if you're having caseating granuloma over there, this will destroy the lymph nodes typically. Okay, but if we have a normal activation of a lymph node, so we're talking about lymphadenopathy, and now we're going to compare activation of a lymph node and a lymphoma. And the one which is important, and it's, let's say, still resembles some kind of architecture, but it's a lymphoma, it's the follicular lymphoma. Okay, so I'm going to compare it to follicular lymphoma and what to look for. In case of a normal reactive lymph node, this is a reactive lymph node. And what's the difference between a reactive lymph node in case of some streptococcus or staphylococcus and compared to lymphoma? So what's the difference? So in case of normal infection and reaction to it, the follicles and granules they get activated, but they will preserve the architecture. So architecture is preserved. Yeah. So this is a reactive lymph node. This is a follicular lymphoma. In case of lymphoma, the follicles will invade the paracortex and medulla. Okay. So loss of architecture. And it's a slow growing lymphoma. Okay. Another difference is that typically what we see during the activation of the lymphocytes and selection of the ones that have good affinity to antigen, we see then apoptosis, and typically we see lots of apoptosis over here. So they are like sort of lacunas, and they're full of the tingible body macrophages, which are destroying the not selected lymphocytes that went through apoptosis. In case of follicular lymphoma, there will be no apoptosis. Okay, there were really low apoptosis, and you won't see active tingible body macrophages. Okay, so no apoptosis. Over here we have apoptosis. Another thing, if you're going to cut the lymph node and you're going to do expression or the qPCR, you will see that there's going to be high expression of BCL2, of course, as we talked about. That's the anti-apoptotic protein. So, or if you're staining for the protein, there's going to be lots of it. Okay. And last thing, over here you should see polyclonality. And there's going to be rather monoclonality because it's a clone of lymphocytes. Okay. So out of this, what is important? If we think it's a infection, acute infection or whatever, and the lymph node is only reactive, or there could be a bacterial, Listeria, whatever, in the lymph node, you can do a puncture and then you can do PCR or whatever and try to find out what is the infection. So you can just puncture the lymph node and it's okay if you want to find out a infectious particle over there. But in case you think it's a lymphoma, what you have to do is a excisional biopsy. Okay, so you have to take the whole lymph node out. Okay, you have to take this. So over here, it's okay to do biopsy only, and just suck something out of it and send it for analysis. But watch out. Of course, if those bacteria, there could be atypical bacteria that are growing slowly, they're intracellular, so many times you won't get anything out of the biopsy. So in this case, and if there is a necrosis happening, you do excisional biopsy as well, and you can send it for panbacterial PCR, or even better, for a specific PCR for the specific pathogen. So if you think if it's this pathogen, you will send it and you will use a specific primers. Okay. So at the end, especially with, for example, Francisella or Listeria or tuberculosis, it's always better to do a direct specific PCR and out of the whole lymph node, because sometimes the bacteria, they stay in special parts, in a stroma, wherever, and you don't have to aim with the puncture for that. So if there's a suspicion it's a bacteria, it's an atypical bacteria, and you're not getting anything out of the biopsy, then I would recommend definitely do excisional biopsy and send it for PCR. Okay. With the lymph nodes that you suspect for any other pathology, cancer, lymphomas, whatever, of course you do excisional biopsy right away, because what you need is the architecture of the lymph node. That's the important thing. So that's why you're taking the whole lymph node out and you put it under microscope to see the architecture. That's crucial. Okay. So that was about lymph nodes and lymphadenopathy. Okay. And we're heading to B-cell lymphomas finally. Okay. So that was introduction, sort of, some terms, and I'm going to go pretty fast through it, but just I'm going to stress some important parts. And sorry, we're going to first do the C leukemias, lymphomas. Okay. Yeah. So let's get to T cells. So thanks for watching, and don't forget to subscribe and ring that bell. And as always, check the description below for supplementary questions and other materials.